Hello students, my name is Monica. I am lecturer of biology posted at Government Senior Secondary School, Bhangala in Hoshiarpur district. My topic is regulation of gene expression. It includes inducible operon system and repressible operon system. The main objective is to create the exact image of protein synthesis at molecular level, understanding the components of operon system, understanding the process of operon system, difference between mode of action of inducing and repressing operons. Introduction. All the cells in the living cells is not active all the time. They become active when needed. Each gene is normally more or less repressed, you can say inactive, but when a particular enzyme is needed, the relevant gene becomes depressed to increase the production of that enzyme. A fully de-repressed gene can produce a fantastically large amount of enzyme. If the synthesis of all the enzymes is fully accelerated, metabolic chaos would ensue in the cell. Thus, the phenomenon of gene expression and de-repression provide a means of decreasing or increasing the rate of synthesis of a particular enzyme in response to variations in the environment requirement for the enzyme. In other words, a cell has a mechanism that controls the expression of a particular gene and thereby the amount of a particular enzyme at a particular time. The rate of protein synthesis is regulated by genetic apparatus and environmental factors in accordance with the need of a cell. The mechanism which stimulates the expression of certain genes and inhibits the expression of others is called control or regulation of gene expression. Overview of regulation of gene expression. Why there is a need of gene regulation. The cells needs to regulate gene expression to economize by not investing energy and resources in synthesis of unnecessary RNAs and proteins when not required. In addition, only expressing a subset of genes in each cell saves space because DNA must be unwound from its tightly coiled structure to transcribe and translate the DNA. Cells would have to be enormous if every protein were expressed in every cell all the time. The control of gene expression is extremely complex. Malfunctions in this process are detrimental to the cell and can lead to the development of many diseases, including cancer. The basic concept for how transcription is controlled in bacteria was given by Jacob and Monod in 1961 by operon model by studying lactose metabolism. It got them Nobel Prize in 1965. Regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at transcriptional level. Formation of primary transcript takes place here. Processing level, regulation of splicing. Transport of mRNA from nucleus to the cytoplasm, and translational level. Regulation in prokaryotes. In prokaryotes, control of the rate of transcriptional initiation is the predominant side for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affects its ability to recognize start sites. In prokaryotes, there are two main types of regulation. Inducible regulation. It operates in a catabolic pathway. It is caused by a new metabolite which needs enzymes to get metabolized. It turns the operon on and it starts transcription and translation. Repressor is prevented by the inducer from joining the operator gene. Lack operon is the example of inducible regulation. It is a negative control. Now, repressible regulation. It operates in an anabolic pathway. It is caused by an excess of existing metabolite. It turns the operon off and it stops the transcription and translation. Aporepressor is enabled by a co-repressor to join the operator gene. Trip operon is the example of repressible regulation. It's a positive control. Students, now let's understand the operon concept. What is an operon? It's a part of DNA that comprises operator and structural genes and functions as a regulating unit in transcription. It works in cooperation with the promoter, a regulator, a repressor protein, and an inducer. The examples are lac operon, trip operon, era operon, his operon, wall operon, etc. The components of operon: structural genes, operator genes, promoter genes, regulator genes, repressor and inducer or co-repressor. Lac operon, inducible system. The word lac refers to lactose. In lac operon, a polycistronic structural gene is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria. In E. coli, breakdown of glucose requires three enzymes. These enzymes are synthesized together in a coordinated manner by a functional unit of DNA, and that is lac operon. Since lactose is the substrate for the enzyme and it regulates the switching on and off of the operon, thus it is called inducible operon system. Regulation of lac operon by repressor is referred to as negative regulation. Components of lac operon. It has the following components. Number 1. Structural genes. Lac operon has three structural genes. LAC-Z that codes for beta-galactosidase, which is responsible for the hydrolysis of lactose into glucose and galactose. LAC-Y codes for permease, which increases the permeability of the cell to beta-galactosidase. LAC-A, it codes for trans-acetylase, which can transfer acetyl group to beta-galactosidase. Number 2, operator gene. It interacts with the protein molecule or regulator molecule, which prevents the transcription of structural genes. Number 3, promoter gene. It provides site for binding of RNA polymerase. Number 4, regulator gene I. The gene codes for a protein called repressor protein. It is synthesized all the time from the I gene. That is why it is constitutive gene which is functional always. The repressor protein combines with the operator gene to repress its action. It controls the activity of the operator gene. Let us now see what happens when lactose is added to the E. coli culture. A few lactose molecules are carried into the cell by the enzyme lactose permease molecules, as a small amount of this enzyme is present in the cell even when the operon is not working. These few lactose molecules are converted into an active form which acts as an inducer and binds to the repressor, a product of regulator gene. The repressor-inducer complex fails to join with the operator gene, which is turned on. The three structural genes are expressed as three enzymes to metabolize lactose to galactose and glucose. Lactose thus acts as the inducer of its own breakdown. When the inducer level falls, having been metabolized by the enzymes, the operator is blocked again by the repressor, so that the structural genes are repressed once again. This is an example of a negative feedback mechanism. The inducible operons generally functions in catabolic pathway. As you can see, in the absence of lactose, the lac repressor binds to a DNA sequence called the operator. It is found between the lac-Z gene and the lac promoter. In this way, the lac repressor blocks the path of RNA polymerase to reach the lac-Z, Y, and A genes. So, the operator remains switched off. In the presence of lactose, lactose molecules are metabolized by the lac enzymes. An intermediate is formed called allolactose. It's an isomer of lactose. Allolactose acts as an inducer by binding to the lactose repressor and changing its conformation. Therefore, it can no longer bind to the operator. Students, now let's see tryptophan, repressible operon. The trip operon is a group of genes that are transcribed together, encoding the enzymes that produce the amino acid tryptophan in bacteria. The trip operon was first characterized in E. coli, and it has since been discovered in many other bacteria. The operon is regulated so that when tryptophan is present in the environment, the genes for tryptophan synthesis are repressed. Hence, it is called as repressible operon. Components of tryptophan. It has the following components. Number 1. Structural genes. Tryptophan operon has 5 structural genes: TRPE, TRPD, TRPC, TRPB, and TRPA, which encodes the enzymes needed to synthesize tryptophan. Operator gene. It lies adjacent to structural gene. It has on and off mechanism. When it is on, structural gene start expressing, and it is off, they stop expressing. Promoter gene. It provides site for the binding of RNA polymerase. Regulator gene, TRPR. It controls the operator gene in regulation with a substance called co-repressor. The regulator gene codes for a protein called aporepressor. The aporepressor combines with the co-repressor and forms a complex called aporepressor-co-repressor complex that joins the operator gene, which is turned off. The structural genes now stop transcription and enzyme synthesis ends. Co-repressor. It is a non-protein compound which may come from outside or from metabolism within the cell. Its concentration controls transcription in the cell. If concentration of a co-repressor rises, aporepressor-co-repressor complex is formed. The complex joins the operator and production of co-repressor stops. If concentration of co-repressor falls, aporepressor fails to get co-repressor and does not join the operator gene. This restarts the formation of co-repressor. In tryptophan operon, tryptophan is co-repressor. Now you can see in this picture, the genes are controlled by a repressor, just as the lac genes are. But in this case, the ligand that controls the activity of that repressor, tryptophan, acts not as an inducer but as a co-repressor. That is, when tryptophan is present, it binds the TRP repressor and induces a conformational change in that protein, enabling it to bind the TRP operator and prevent transcription. When the tryptophan concentration is low, the TRIP repressor is free of its co-repressor and vacates its operator, allowing the synthesis of TRIP mRNA to commence from the adjacent promoter. Surprisingly, however, once polymerase has initiated a TRIP mRNA molecule, it does not always complete the full transcript. Indeed, most messages are terminated prematurely before they include even the first TRIP gene, TRIP-E, unless a second and novel device confirms that little tryptophan is available to the cell. Students, let's see the animation video to understand the concept of gene regulation.
All organisms, large and small, need some way to control their gene expression. Otherwise, they would not be able to control what proteins they're producing and when they're producing them. This would lead to a lot of wasted energy and resources in the cell. Being able to control when genes are expressed based on when their products are needed helps the cell be as efficient as possible. In prokaryotes, one way to control gene expression is through the use of operons. This is your basic operon structure, a series of structural genes that share a promoter, an operator, and a termination sequence. In addition to these parts of the operon, a key player in the expression of these structural genes of this operon is the regulatory gene, in this case located upstream of the operon. The promoter of the operon is where RNA polymerase will bind to initiate transcription of the genes that follow. You can think of this operator as the stoplight for RNA polymerase, either allowing or preventing RNA polymerase from transcribing all of the structural genes. All of these structural genes are linked to the same promoter because they encode proteins involved in the same metabolic process. You can think of them as members of an assembly line, each with an important job in creating a single end product. There's not an instance where you would need one gene expressed and the others not expressed because they are all needed in the same pathway. It's simply more efficient for them to all share a single promoter and operator. The genes are either all expressed or all repressed. And then you have this regulatory gene. This gene encodes a repressor. As you can see, this repressor has a DNA binding domain, and this domain fits onto the operon's operator perfectly. Environmental factors can interact with the repressor to make it active or inactive. When the repressor is active, it will bind to the operator. When it binds, it can prevent RNA polymerase from binding to the promoter and transcribing the genes, effectively limiting structural gene expression. When the repressor is inactive, the structure of its DNA binding domain is altered so that it cannot attach to the operator DNA. Because the inactive repressor can't bind to the operator, RNA polymerase can effectively attach to the promoter and transcribe the genes. In this circumstance, the operon is expressed. So changes in the external environment or changes inside the cell can make a repressor change shape, allowing the cell to adjust to changes by making only the proteins necessary in the moment. The most common example of an operon is the lac operon, which contains structural genes that encode enzymes that help break down lactose. When lactose is absent from the environment, the repressor is active and the lac operon is repressed. This means that the structural genes are not expressed. This prevents the cell from making proteins to break down lactose when it doesn't actually need them. When lactose is present, the repressor becomes inactive and the lac structural genes are expressed, producing proteins necessary to break down the lactose in the environment. In this way, the presence of lactose induces the operon, leading to lac gene expression. This is your TRIP operon, which we find in the DNA of bacteria E. coli. These 5 structural genes encode proteins necessary to produce the amino acid tryptophan. And the TRIP-R gene here encodes an inactive repressor that, when active, engages with the operator here to limit transcription of the structural genes. When there's a lack of tryptophan in the cell needs to make more of it. The repressor is in an inactive form, and the RNA polymerase can bind to the promoter of the operon and transcribe the TRP structural genes, leading to the production of tryptophan in the cell. When there is an abundance of tryptophan, the cell doesn't need to make more. In this state, the tryptophan will act as a co-repressor, binding to the repressor and causing it to become active. In this state, the repressor will bind to the operator and prevent RNA polymerase from transcribing the TRP structural genes. Here's the kicker. When TRPR is mutated and the cell is not making a functional repressor, you'd expect these structural genes to constantly be expressed, but that is not the case. Cells with no TRP repressor will still limit transcription of these structural genes when there is an abundance of tryptophan. This indicates that there is another mechanism at work to help limit tryptophan production. That secondary mechanism is called attenuation. Attenuation means reduction of force. So in this case, the TRP operon attenuation means limiting the amount of transcription of the TRP structural genes. And the whole process of attenuation has to do with this sequence here between the operator and the structural genes, which is called the leader. When RNA polymerase binds to the promoter of the operon, the first thing transcribed is the RNA leader. Structural gene expression is controlled by this RNA leader, which in certain circumstances can cause transcription to be terminated before RNA polymerase reaches the structural genes. Now, this leader sequence has some regions of self-complementarity. Highlighted 1, 2, 3, and 4 here. When RNA has regions of self-complementarity, it will often fold in on itself to form stable structures. There are 2 stable conformations for this RNA leader sequence to form. Options for stable conformations of the RNA leader. Let's focus over here. This formation contains 2 hairpin loops, and this one is called a terminator. When this hairpin loop forms between regions 3 and 4 of the RNA leader, it causes transcription to stop before the polymerase can reach the structural genes. If the RNA leader forms this structure, the structural genes are not expressed, meaning the cell will not be able to make tryptophan. So the RNA product of transcription in this case is shortened, attenuated RNA with no structural gene sequence. Alternatively, when we see this formation, where the region 2 and 3 are bound together, we see this single hairpin loop form. This hairpin loop is called an anti-terminator. It will not interfere with the transcription of the structural genes, resulting in a longer RNA sequence produced by RNA polymerase, including the structural gene sequence. So the TRP structural genes can be expressed and tryptophan will be produced by the cell when the RNA leader creates this formation. So what causes one RNA leader conformation to form over another? Just like we saw with the work of the repressor protein, the deciding factor is the level of tryptophan in the cell. When there is less tryptophan, you can see this conformation containing the anti-terminator, which allows the cell to make more tryptophan. When there is an abundance of tryptophan in the cell, you can see this formation containing the terminator, which prevents the production of additional tryptophan through TRP gene expression. So how does the presence or absence of tryptophan dictate the shape of the RNA leader strand? As a reminder, in prokaryotes, translation can start on RNA that is not completely transcribed yet. So as the RNA polymerase is completing transcription, a ribosome can attach to the RNA already produced and start building protein. This is called coupled transcription translation. The key to this whole attenuation process is the sequence right at the beginning of the leader. Early in the leader sequence, you see an open reading frame that contains 14 codons, 2 of which are codons that encode tryptophan. The speed at which the ribosome will move through this small section of RNA depends on how much tryptophan is available. If there's an abundance of tryptophan, then the ribosome will be able to move quickly through this section, using the abundant tryptophan readily in its translation process. When the ribosome moves quickly, this allows the formation of the terminator hairpin loop, resulting in the attenuated RNA sequence. When there is less tryptophan available, the ribosome stalls at the 2' tryptophan codons, taking longer to move forward with translation of the RNA as it waits for tryptophan to be incorporated into the polypeptide. Because of this stall, the anti-terminator forms, allowing transcription of the structural genes and expression of the proteins that will help the cell make more tryptophan. So to review, when there's a lack of tryptophan in the cell needs to make its own. The slower movement of the ribosome as it translates the RNA leader open reading frame allows the leader RNA to fold into the conformation that includes the anti-terminator, which in turn allows the transcription of the full sequence of the TRIP structural genes, which will then be translated into proteins that will help build more tryptophan. When there's an abundance of tryptophan in the cell and the cell doesn't need to express the structural genes, the ribosome will move quickly through the translation of the leader RNA, and this quick movement leads to the creation of the conformation of leader RNA that includes the terminator hairpin loop, which causes the transcription of the TRIP operon to end early before the structural genes. The production of this attenuated RNA that does not include the TRIP structural genes means that the cell will not produce the proteins necessary to make tryptophan. This complex system that includes both the RNA leader and the TRIP-R repressor allows the cell to adjust TRIP operon mRNA production according to the amount of tryptophan in the cell, meaning the cell won't waste energy making something that already exists in abundance. You will see similar attenuation mechanisms in other operons, including histidine, leucine, phenylalanine, and threonine operons.
Thank you for watching the video.